1 out of every 4 Canadians will suffer a stroke in their lifetime. Now, be honest, when I say the word stroke, what do you picture? I'm going to take a guess and say you thought of someone elderly. But did you know that stroke can happen in young adults, children, and can even occur before birth? Now, to better picture what a stroke is, I want you to imagine a garden hose. When there is a kink in the hose, there will be no water supply for the garden. On the other hand, if there is too much pressure in the hose or it's made faulty, it can leak or burst. The same thing can happen when blood is trying to get to different parts of your brain. When this blood flow is interrupted or reduced, it prevents brain tissue from getting the oxygen and nutrients it needs and can lead to devastating damage. This blood is delivered through a network of microvessels called capillaries spread throughout your brain. These small vessels are supported by cells called pericytes that reinforce those vessels, helping them develop and manage that blood flow that is constantly coming through. One of the diseases where we see leaky or bursting vessels is cerebral small vessel disease, or SVD. This disease weakens the smallest vessels of your brain and is a contributing factor in up to 25% of strokes. Now, despite the prevalence of this disease, we don't really know why or how these vessels become weak, and there are no effective treatments for those that suffer. What we do know, however, is that the genes a person is born with play an important role. One of these genes is called FOXF2. Patients with FOXF2 mutations suffer from SVD and have a higher risk of stroke. In my lab, I study precisely how mutations in FOXF2 affect the small vessels of the brain from the moment development begins. I do this by using a unique FOXF2 mutant zebrafish that we created. So far, I have found that my mutants' brains are adversely affected early in their development. They suffer from more hemorrhages and have a profound loss in the number of pericytes, sometimes even hemispheric loss. While some of these mutants may recover enough to survive to adulthood, a significant portion of them never recover and are left with compromised vasculature. But what exactly determines these two trajectories? New emerging data from our collaborators have shown that the hallmarks of SVD are present in the brains of asymptomatic young adults as early as their 20s. Movement in the field is demonstrating that the answer may be that stroke is rooted in development. I have a developmental disease model in which we can look at the underlying mechanisms related to FOXF2 mutations to better understand how stroke risk develops over the lifetime. We have already uncovered novel information related to vascular stability in our stroke models, and the work has only just begun.